Hello everyone, hope you're doing well. We're going to get started here, I think pretty quickly today. Sorry I'm running a little bit behind. Wanted to make sure I had all the figures that I needed in place to show you because we're going to have some illustrations of some of the things that we do. But welcome, thanks for joining us. Feel free to say hi in the chat, especially if you're new. Let us know where you're coming from. Sarah, hello, welcome. Henry, glad you can join us. We are going to be covering assembly too. I hope it's not a sticking point. I think I'll give a refresher of some of the concepts that we looked at in a previous stream where we started talking about genome assembly, but more on that here momentarily. Tammy, welcome from London. Great to have you here. Yes, we're going to talk a little about assembly today. And we're going to look at 5 problems, all related to genome assembly. And these are good questions. The point is, I think we're only really dipping a toe in the waters of genome assembly. It's a challenging area that people have worked on algorithms for assembling genomes for literally 30 plus years. And it's still an active area of research, although we have really good genome assembly software these days. We'll see kind of a sampling of some of the ideas of genome assembly. So you at least get, the challenges here are not trying to get you to make, say, real software around all of the different considerations that you might have to keep track of with practical data. But I think that the idea here is to give you a sense of what those challenges are. And in some cases, I think the problems that we look at isolate these. I'll also mention it as we go through this, but these are challenges that we wrote pretty early on. And special thanks to Misha Dvorkin and Ryan Chicky, who actually created a few of the problems that we're going to be looking at today. But since that time, we have written an entire textbook that's now in its 3rd edition. We need to make a 4th edition of the book. But if you're interested in kind of a 70-page long treatment of that work, you can check our work out at bioinformaticsalgorithms.org. So we have a print book associated with this. We have an interactive textbook associated with it. And join our mailing list and Discord because, I'm going to be teaching this Great Ideas in Computational Biology course here coming up late summer, early fall that will get into some of the algorithms for genome assembly as well as genomics and topics beyond genomics. But we will talk more on a theoretical level without doing a lot of coding, because when you really try to code algorithms for genome assembly, you're going to get very, very deep very quickly. It's going to be hard to know where to stop. So this is on the coding level. This is kind of a sampling of that. But there are also some beautiful mathematical ideas, and I'll say a little bit more about that when we do our first question, because these problems in genome assembly are the reason why I entered the field of computational biology, why I really wanted to get connected to this field on an educational level. But I'll say a little bit more about that momentarily. And otherwise, I think we're about ready to get started. So we have starter code and completed code in the description of this video if you would like to follow along. And I always say too, if you have questions about what we're doing, feel free to ask those. Usually what I'm doing with these streams is I'm going through an entire solution to a problem, or hopefully solution. We're 10 episodes in, we haven't had any major pitfalls as of yet, but I'm walking us through the solution to the question. And then if you have questions, usually I'll stop at the end and between problems and take a little break and point out things about code if there's anything that you're curious about. So let me know. Otherwise, we're about to get started. Yeah, Henry, welcome from Brazil. Excellent. Okay. As always, of course, like and subscribe. Drop a comment here to our video. Ask questions in the chat. All that stuff is super helpful. Keeps us visible. And yeah, we love to see it. So let's get started. And we're going to be looking at DBRU first. So I have some starter code, and as always, what we've done before is, I've got our file explorer, and it has all of the files as well as an updated custom io.py. This continues updating as we go, and then each file corresponds to one problem. So my file explorer here is in VS Code and contains all the problems that we solved, and we're going to start with DBRU. Okay, so we can begin. DBRU is constructing a Bruijn graph. And this is a network that I really love. Before I talk to you about what it is, I will say that we've started discussing genome assembly already. So we looked at a previous problem, and that previous problem that we looked at was this long ID. And the figure that we showed in that particular context is a common type of figure that you see very frequently, which is, centrally throughout the age of genome sequencing, the central question is that you have many copies of a chromosome, or many copies of a genome, which is divided over multiple chromosomes. That biological sample is easy to obtain. We can fragment it down into all possible fragments, maybe of variable lengths, maybe of equal or approximately equal lengths. And then we need to use overlapping information in order to lay out those reads and then be able to construct or reconstruct the original genome. So it's an old picture. This paradigm of what we're doing goes all the way back to Sanger sequencing in the late 70s and being able to sequence a bacteriophage, or a virus that attacks bacteria that only has a genome that's like 5,000 nucleotides long. But even though that's the case, if you want to assemble a human genome, the picture is largely the same, although these days we also have a database of more information. So we have genomes from a lot of different humans, and when you have that sort of reference context, it changes the picture a little bit here. It's not as if we go in blind and we just say we're fragmenting this down and assembling everything. So in that particular context, the algorithms that we're going to use are going to take a slightly different trajectory, but we're going to work on these problems here with what we would think about as de novo genome assembly. And that's just a fancy term for saying we don't really take any other information into account when we're solving the problem other than what the sequence identity of these fragments are that we fed into a genome sequencer. So the genome sequencer can't read an entire chromosome at a time, but it can read these short fragments. And at the end of the day, when we sequence these fragments and know their identities, we need to overlap them and figure out what the underlying genome was. So we've worked a little bit with genome assembly in this particular context, although we had a very straightforward question where a greedy algorithm worked. So the fragments overlapped by a lot, and we were able to assume that every fragment overlapped with only one other fragment, as well as the fact that they're all from the same strand of the chromosome. Now, in the case of real DNA, when you fragment the DNA, you're going to be reading one strand or the other. There's no real control over that process. So you may be reading DNA from one strand, or you may be reading it from the opposing strand, and we need to take that into account. Up until now, we kind of assumed away any issues. We said there's no errors in the reads, there's no double-strandedness, and we have tons of overlaps. Everything is going to be perfect. And we want to come back and generalize this question a little bit and see, at least get a flavor of the types of approaches that we might use when the problem gets more challenging. There are, of course, a lot of challenges in genome sequencing and genome assembly, which is why it's been an active area of research for decades. And of all of the considerations of this problem that make it practically complex, the number one most complicating factor is not errors in reads. It's not the fact that DNA is double-stranded. It's that the problem is that you have so much repeated information. And I'll show you what I mean on, it's always good to see an analogy of this sort of thing and think about this problem in the context of solving a puzzle. And if we look at the world of jigsaw puzzles, a natural connection is what happens when a jigsaw puzzle starts having repeated information. So in the 90s, there were these triazyl puzzles that were quite popular. You could find them in gift shops around the US. And they all had the exact same shape. They were 16-piece puzzles where each piece had the same triangular shape. And I think the tagline on these was, it's harder than it looks, because it's only 16 pieces, how hard could it be? And what made the puzzle challenging is that there was repeated information. So what you can see in this particular case is there are, I believe, 4 red frogs, but they're all divided in half. So each piece might have the left or the right side of one of the red frogs. And that means that this left side of the frog could have gone here, but we didn't know that necessarily. Like, it might have gone here as well. This right side of the right frog could have come here, or it could have come from this piece, and so forth. So you get this kind of an exponentially growing number of possible solutions because of that. And you don't just have, one frog with repeated information. It's the entire puzzle is this way. So you have multiple frogs with repeated information. And so if you apply a brute force algorithm, what will happen is you might get 13 pieces in, and then the last 3 pieces don't make sense. And so it would be nice if somehow we organize the data in advance, so we're not just brute forcing the algorithm, and so we're not just kind of hunting blindly for a solution. I really enjoy a version of this puzzle kind of taken to the extreme, which here is shown, at least at this point in time, the most challenging jigsaw puzzle ever produced. And one that comes with that at one point in time came with a $2 million cash prize for anybody who solved it correctly. So it's by far the highest stakes jigsaw puzzle ever assembled. And it does have a solution. Actually, it has many solutions. But between 2007 and 2010, the community was given as much time and as they needed for like a 3.5-year period to solve this puzzle with a deadline. And after 2010, New Year's Eve came and went, and no one had solved the Eternity 2 puzzle, even though people had access to supercomputers, and even though this puzzle only has 256 pieces. It's applying the same principle, though, of the same information repeated multiple times throughout the puzzle, and it borrows this triazyl idea of dividing each pattern in half and then repeating that throughout the puzzle. So everything here is consistent in that you can see everything is getting assembled, and when you're here, you're looking for a piece that has this orange pattern on one side and this pink and yellow one on the other. You might be able to see a piece that satisfies those conditions, but the problem arises in the same way. You might get 2/3 of this puzzle assembled, or 3/4, and eventually you hit a stopping point because you're actually nowhere near a solution. It might seem like you are, but the problem is that the number of candidate ways to start this puzzle, and it seems like it's correct, is astronomical. It's growing exponentially so fast that even though you're at 256, you have, I think, far, far more candidate ways of putting this puzzle together in a reasonable way than there are atoms in the universe. But you're looking for, a tiny collection of atoms, like in the bracelet of a cat or something on one planet. And so any kind of brute force approach to this is simply not going to work. And unfortunately, there really isn't a faster way of going about organizing this solution. So from that perspective, it might seem like genome assembly is impossible, because this is the exact same issue that we face when we assemble genomes. About 50% of your genome and my genome consists of repeated fragments. And that's going to create an issue, because if we're thinking about overlapping fragments, well, you might have multiple different candidates at every point in time. It's analogous to saying I have a puzzle piece. Well, I could put this in a lot of different places. And our only hope, especially because in the case of a human genome, we're going to have hundreds of millions of sequencing reads. Our only hope is to organize those reads intelligently before we begin, and so that somehow we might be applying a more advanced algorithm to peer into the structure of this data. So I'm not just picking up a piece and saying, oh, where does it match over there? That is an approach that is guaranteed to fail. And there are going to be two ideas that we can apply to do this. In either case, what we're going to do is establish in either case, we're going to organize our sequencing reads in some way into a network. And the reason why we do that is because that's what we always do in biology. Biology is a science that is all about paired connections, paired relationships. And that means that networks are everywhere in biology. For example, you might have a network where all of your nodes are proteins, and you connect two proteins with an edge if they interact in some way. If there's a known interaction between them, perhaps as verified by laboratory experiments, you get a massive network. Or your network might be proteins again. Some of those proteins are special proteins known as transcription factors, and you connect a transcription factor to all of the proteins that it's known to regulate. In this particular case, we have another type of pairwise relationship, although we're going to connect it to an algorithmic problem. The pair relationship that we have here is you have sequencing reads, so it makes sense for those to be our nodes. And we connect two nodes with an edge if they overlap. We can be a little more precise about that. So, for example, if all of your reads are trimmed, for example, down to a uniform length k, well, you'll notice we connected CCA to CAT. Why? Because they overlapped in k-1 symbols. All of the edges here are one directional, so we don't go backwards, because CCA overlaps with CAT in the CA, but CAT doesn't overlap with CCA. So in this formulation then, what we're trying to do is find a path, a collection of nodes that are connected via these edges. And that path should be using every single one of our nodes, meaning it should touch each node. And as we walk through, well, we can start assembling the genome. So we would say, when we're at this node, we are TAA, and then the AA overlaps, so we would have a T. And then if we went this direction, we would have a G. We might bounce over here and add another G. So there's multiple different alternatives through this. And then we go, over this way, and we have TGG and GGA, so we add an A, and so forth. But the idea is that at the end of this process, we want to have a path that touches every single one of those nodes. That way it will explain all of our sequencing reads. And then we can infer the genome that comes from that. The problem with that approach, though, is that type of path in a network is called a Hamiltonian path, a path that encounters every single node exactly once. And the problem is that, well, no one has ever invented an algorithm that is efficient for finding a Hamiltonian path. This problem is NP hard, which means that it is unlikely that we're going to get a fast approach for it. And it's one of these problems that sits at one of the biggest unsolved questions in all of mathematics and computer science. Rather than hoping that we can solve an unsolved problem in computer science, what we will do is look at a different network. And in this network, we take the same information. We have a collection of sequencing reads. They're the same reads that we had in the overlap network previously. But in this particular case, we're assigning the reads, or the fragments of DNA, to edges. And when you look at an edge like this, here CAT is a sequencing read, but it's connecting CA to AT, so we connect its prefix to its suffix. And if we were walking through the network, well, imagine that we're here, we go TG to GC, that's sequencing read TGC. When we look at the next read, it's GCC. We don't see it. You're right, I don't see it either. Give me a second. Let's try that again. There we go. That should work. Let me know if you can see that now. Could you not see the previous network? I hope I wasn't just explaining explaining something with no figure. Very occasionally this happens, that you just get a freeze on the share, but we haven't had that happen in months. Okay. Okay. So I was just explaining figures that weren't shown. Do you want to see the figures? That's amazing. I can't believe anybody was here. You must have thought I've gone mad. Okay, you probably should see the figures. All right. Here's our triazole puzzle. There's our frogs. Okay. Yeah, I mean, I guess I'm just that good at explaining things that you can just conjure up what I'm talking about. Here's our triazole puzzle with frogs. And you can see the different frogs there. Here's our eternity puzzle. All right. And in the eternity puzzle there, you see the 256 pieces. You can see that this has started to be assembled correctly. You have an orange fragment here and a pink and yellow one there. So we're looking for a piece that fits in there. You'll get most of this done, and then there's just going to be a point at which you're stuck and nothing else fits. And what else do we have? We had our overlap network. So our overlap network here was taking pairwise relationships and connecting them. Okay, so our pairwise connections in this particular case are nodes connected to each other via edges if they overlap. So for example, I was saying AAT connects to ATG, and what we're trying to spell out is a genome that has all of these nodes involved in it. So as you go from TGC to GCC, for example, you overlap in the GC in each of these positions. So you would be adding on one extra C. And then we made this transition to a de Bruijn graph, because the problem with that formulation is if you're trying to find a path that touches every node in a network exactly once, then that's not going to lead you to a solvable problem. However, in this particular case, we've assigned the sequencing reads to edges. And if we walk in this direction, you see you have TGC, and the next one overlaps by quite a bit. So GCC, and then the next one overlaps in K-1 symbol. So the CC overlaps, we have CCA, and so on. So you're still able to walk through this network and touch all the different edges that we see and assemble a genome by taking overlaps. But as we go through this, the point becomes, well, what was it that we need to find? Here we need to assemble the genome by touching every edge exactly once. And there's a critical division here, which is that this is a special type of path in a network called an Eulerian path, which uses every edge exactly once. This problem has a known fast solution. And there's a big mathematical history here that goes back a couple hundred years that we're not going to take the detour to talk about right now. But this is why I really kind of came to this field of computational biology education in the first place. Because, well, that mathematical story is a story that I knew well, but it's not something that I ever anticipated in my wildest dreams showing up in the context of biology. I had no idea that there was anything algorithmic of interest in the field of biology. I thought it was just dissecting frogs and memorizing things. I, of course, was very wrong. And we're now ready to take a look at, how is it that we would solve this problem? And the first thing we might want to do is make this de Bruijn graph. So we're going to make this de Bruijn graph here for a collection of nodes or collection of reads. And so this problem is going to say our input is just a bunch of DNA strings, and we've presented it in a very fancy mathematical manner. But ultimately, what we're going to do is make this de Bruijn graph where the sequencing reads get assigned to the edges, and then the nodes are the prefixes or suffixes of those reads. When we do that, we're also going to do one other thing, which is to add a little bit of the biological knowledge here, and that's to take the reverse complement of every one of our sequencing reads. So we're not just going to take each read, we're also going to take its reverse complement. And the reason why is, if I have this sequencing read and we're trying to assemble one strand, well, the problem is, we could be on either of the strands. And so we're kind of bolstering the amount of information we can infer. If I have a sequencing read, I also am presumably inferring that it lives somewhere on the reverse complement, or its reverse complement lives on the complementary strand. And so we're going to make this de Bruijn graph, but we're also going to let the edges not just be the input sequencing strings that we have, we're also going to add their reverse complements to it. And at the end of the day, we're going to take an adjacency list that corresponds to that. So what does that mean? Well, in this case, this means ATC to TCA and ATG to TGA. These pairs are going to be the pairs of nodes that each edge connects. So when we look at this, we say, well, I don't see an edge corresponding to ATC TCA, right? It's not present in the data set. There are other places where we might. Okay, so in this particular case, CAT to ATG. Well, you see that they overlap in the AT. I've frozen again. Goodness me. I really want to solve the problem, but if you can't actually see my screen, we're going to be in rough shape. Huh. Okay, give me a second. Okay, let's give that a go. Sorry, we had unbelievable technical difficulties, and we lost like the whole setup of the background in the middle of that too. So we will try this again. I'm actually just going to start from the top, and we lost some of my explanation. If we get more freezing like this, we'll just shut down the stream for today, and that's okay, and we'll give it a go next time. So in the meantime, I am going to give this a try. Let's see. Hopefully you can see what we're working on. But we're working on question DBRU, which is constructing a Bruijn graph. And in this particular case, we're given a set of input DNA strings. And when we look at this collection of DNA strings, we're going to produce an adjacency list of a network. That's this Bruijn graph, where we take all of the strings, all of their reverse complements, and then we're converting them into edges. So if we look at a particular edge like CAT to ATG, this appears in the data set as CATG. So CAT represents its prefix of length k-1, ATG is its suffix of length k-1. So that is the edge that we see in the data set, CATG. There's another place, ATCA is what we would infer from this particular string, but we don't see it in the data set. And we'd say, well, where does this edge come from? If it is the edge ATCA, then when we reverse that and we take its complement, so we take the reverse complement, we get the string TG AT. And that is a string that does occur in the data set. So what we're going to do is we're going to take all of our strings in the input, take them and their reverse complements, and when we do that, we're going to form a bunch of edges connecting prefixes to suffixes, and that's where our sample output comes from. Okay. Now, we want to convert this to code. And the solution here is we're going to produce two functions. So the two functions that we're interested in are, let's make the Bruijn graph that doesn't necessarily take into account reverse complements. So if we look at an example, and that example is for another collection of strings, we could have a Bruijn graph that looks like this, where our edges are reads of length 3. They connect prefixes to suffixes. And when we look at that, well, this doesn't necessarily take reverse complements into account. So this would be a single-stranded genome, and you can reproduce it by starting at TA, walking across, walking up and around, walking down and around, and then walking across. So we would start with TA, we end with TT. This doesn't include anything about the opposing strand of DNA along with it. What we're going to do is add a version of this where we make the Bruijn graph that also includes this particular case of taking reverse complements. So we'll have two functions, right? We can finally take a look at our code, and we have two functions in our starter code. We have a Bruijn graph function that takes a collection of k-mers and it produces a network with an adjacency list here. Or we could also take reverse complements. So when we take our k-mers, we're going to reverse complement those and then produce the adjacency list. Now, when we say adjacency list, the type of both of these that we're going to have as outputs are going to be a map or a dictionary that maps strings, which are our k-mers, to a list of other things that they're connected to. So this could even include duplicates. Like imagine that it may be the case that I see the same edge or read more than once, which means I'm going to have a multi-graph connecting that node to the other node in multiple places, and that's okay. So this formulation will allow me to represent that. And if I'm given a collection of k-mers and I just want to make this de Bruijn graph of them and their reverse complements, well, the first thing I want to do is make a combined list. And that combined list is going to start off, I'm going to call it combined. It's a list of strings. It starts off as empty. And then what we want to do is range over all the k-mers and add in themselves and their reverse complements. So I've already imported reverse complement from the problem REVC, which we solved long ago. And all I need to do now then is to say range over all of the input strings and append two things. We're going to append S, and we're also going to append the reverse complement of S. And what's nice about that is then I've grown the collection of k-mers. And so now all I have to do is make the de Bruijn graph of all of them. And now I only have to really write one function that takes a collection of k-mers, and that produces their network. So I will return the de Bruijn graph of the combined collection of strings. So we can have two functions for the price of one here. And really what I want to write is our function that takes a collection of k-mers as input and returns this adjacency list, mapping strings to bunches of other strings as we need. Okay, so then it's just a matter of writing this de Bruijn graph function, which is not too bad. We're going to make the graph that we care about. So it is a dictionary of strings to lists of strings, and it starts off as empty. And then, eventually that's what we're going to return. And in the meantime, we want to say, well, let's range over all the k-mers and identify the edge connecting prefix to suffix. So when we look at our sample, for example, and we talked a little about this before, each one of these edges comes from connecting the prefix to the suffix of one of our input strings or its reverse complement. So in the case of ATGC, we said, well, that's connecting ATG to TGC. In general, we're going to need to take it as well as its reverse complement and connect its prefix to its suffix. So we've already done that reverse complement though. And so in general, this is just taking the collection of strings and figuring out what are their prefixes and suffixes. And that's just basic string manipulation. So I'll range over all the k-mers and I'll say that the prefix that I care about is S of everything up to, not quite length of S, but it's length of S minus 1. So it's every symbol except for the last one. And then in the case of suffix, well, it's everything that starts with 1 and then goes to the end of the string. These are the two nodes. So I connect them. And here's where I'm going to say, well, there's something to keep in mind, which is this the first time I've seen the prefix? And if it is, I need to make a list for it. So if the prefix is not in graph, so it's not a key, well, I want to create an empty list for it. And then regardless, at this point in time, now I know that there's a list associated with the prefix. And so what I'm going to do is to say graph of prefix dot append suffix. So this corresponds to adding the edge. And now the graph is complete. And then it's just a matter of running this. So I can see it, hopefully everything is okay. I can run Python 3 DBRU.py, and that runs it on the test data set. And this appears to be, has the right format and everything looks to be in order. Okay. And then it's just a matter of saying, let's comment out the parsing part of the code. And we're going to get a data set from the Rosalind platform. So I'll download a data set. And as always, I'm going to place that data set into my file explorer and just call it dataset.txt and let the parsing handle itself. So when I rerun this, it's going to run the sample, but it'll also run it on the full data set. It'll give me the output as output, and then I can go back and copy that in and submit. Okay. All right. Boy, that was dramatic. That was a dramatic for what was probably supposed to be like a 5-minute exercise. But we got through it. That's what's important. All right. There was actually a good question about, like, what if a string is its own reverse complement? And notice I didn't use a set anywhere, right? Which might be something that you do, but we're not in favor of like throwing out or compressing data. Like that could be a useful case. So when we take the reverse complements, we're just reverse complementing everything. Where were we here? Ranging through all the k-mers, and everything here is an append. So we just have a bunch of lists. And so when we do this, we're appending the reverse complement. And if something is its own reverse complement, well, or it's connected to itself or something like that, none of that is not going to be included by this process. Okay. All right. We're going to move on to PCOV. So that's our next one. All right. PCOV is genome assembly with perfect coverage. And we've learned about what a de Bruijn graph is, and we have a good sense of how this data structure might work for organizing our reads into a form that might lead us to a solvable problem. This particular question that we're going to look at is going to assume that, like is the case with bacterial chromosomes, that the chromosomes themselves are cyclic. So at the end of the day, we're returning a string. We want to form this DNA string, but we also want it to be a cycle. And as a result of that, that means when we look through the de Bruijn network, what we're really looking for is not a path that uses every edge, but a path that uses every edge and returns to its starting point. So if we look at this sample de Bruijn graph, if this were a cyclic string at the end of the day, what we'd be looking for is a path using every edge exactly once, and you can see it's numbered off here. As we walk through, we add each new symbol that we encounter. So we start with TA, A, and then when we go here, we have an overlap with the AA, so we add a T, then we add a G and a C, and so on. And we walk through the network. Notice that every edge is labeled by some number, so we're capturing every edge. And as we capture every edge, we also return to our starting point. So at this point in time, we would be appending to the string, and we would say TGT, and then we'd add another T. But then when we come back in this direction, that edge connecting this to this would be TTA. So when we say TTA, we actually are back at the starting point. So instead of continuing to append, we've wrapped all the way around, and we're back at TAA. This process is only going to work because every time I encounter an edge or a read, the read that overlaps with it perfectly is just downstream of it. So in other words, we're able to, when we go from here to here on edge 8, edge 9 is available to us because it was detected in the sequencing reads. So we need it to be the case that when we capture all the DNA, we're getting every possible k-mer from the sample. And that assumption is quite strong, but it's called perfect coverage. So it's always nice to start with strong assumptions, see what solutions we can obtain, and then use those solutions to try and broaden them. This problem is also going to assume that when we form the de Bruijn graph, let's focus ourselves on assuming that the reads come from only one strand of DNA. So, for example, this might be the case for RNA viruses like coronaviruses, because you only have one strand of RNA that then in the sequencing process gets converted to DNA, but all of your fragments come from just one strand of DNA. We can always generalize that later, but what we obtain in this particular case is an input that's just a collection of k-mers and an output which is an explanation of those k-mers that's as short as possible, meaning we want a candidate cyclic chromosome that contains all of those input k-mers. And you'll notice this looks like a really short string, right? But you can see, well, in this string named Gattaca, we can see GATTA, we can see ATTAC here, we can see TTACA is present, but then you're also going to need to have TACA, and then the G, which we see present here, and then the ACAGA, which wraps around and is present here, and so forth. So we're getting all the k-mers, but we're getting all the k-mers as a circular string. And if we're given these strings, we want to produce this particular string as output. Now the autograder is not going to mind if we start at the G or if we start at the A or the T or the T or the A, the C, the A. It doesn't matter. It will count all of these solutions correctly, but we need to take what we've already learned and apply it here. Now, we're also making a very strong assumption, which is that I told you in a previous problem that this is complicated by repeats. So if you have repeats, you're going to see the exact type of scenario that you might see in this particular cyclic type string that we saw, where when you enter a node, you have multiple options sometimes on where you can go. So when you enter this TG, for example, I maybe could go this direction, I could go down, or I could go over to the right. And so because I've got all those options, that's going to complicate the problem, and I need to make sure that I walk through the network in an intelligent manner. So we talk in our bioinformatics algorithms textbook, you can check that out, because we talk about like how is it that you organize all of this and know which direction to go, or even if you do it blindly, allow yourself to walk blindly and then consolidate this information into a path that's going to touch every edge. However, in this particular case, what we can assume, what we're going to be allowed to assume, is there's only one simple cycle in the de Bruijn graph, meaning I'm really just finding one cycle that's going to walk, starting at some position, walk through the network, and come back. So the algorithm that I'm going to need for this is not going to be as complex, even though this is a useful network. It's not going to be as complex as what it could be in the case where we have a lot of repeats and the network has a lot of tangling. So I have a function here called assemble circular that's going to take a list of strings as input. It's going to return a circular superstring that contains all of those k-mers as input, but that doesn't contain any additional characters. And the way in which we're going to solve it is to build the de Bruijn graph, but then kind of greedily walk through it. So the idea is build the de Bruijn graph and then greedily walk through it until we use all edges and form a cycle. So the first thing that we should do is to say, well, I'm going to form a de Bruijn graph here, and in this particular case, I could even call the function to make the de Bruijn graph itself, I also really could probably do this without making the network entirely, and that would be a direction I could go too. So I will, in this case, form the de Bruijn graph just so I'm comfortable with working with it. I've written the function. I've imported it from DBRU, so that's good. And then once we have built the thing, then it's just a matter of, start at the first k-mer and walk greedily. So I want to say our starting node is equal to go in and access the k-mers. And it's easy to say, well, it's the initial k-mer that we see in our data set. But I'm really starting at the first node. And remember, the first node is a prefix. So it is a prefix starting at, so the initial index is 0. But it goes up to whatever the length of this k-mer is, length of k-mers of 0 minus 1. So that's our start node. And I'm going to set the current node equal to the start. So they're going to refer to the same thing. And then we will use a list of characters to build up the string. And that's just because repeated string concatenations in Python and in a lot of languages can be inefficient. And so we'll say chairs is a list of strings. It starts off as empty. And I'm going to use a parameter n to represent the number of k-mers that we see. And then I'm going to say, well, let's range over all of the k-mers. So range over all the k-mers. And what we will do is append the current symbol to our characters and then move to the next. I'll just say range and move to the next node in the network. So this will allow us to walk. I know how many steps I need to take, which is n. But I'm not ranging over the k-mers themselves. I'm just taking n steps. So, when I move to the next node, I'm going to update current to be equal to the graph of current. And graph of current is a list of k-mers. And that list in general could have a huge number of strings in it, right? Because if I'm at a node, it could be connected to a lot of different places. But remember that in this problem, we're assuming it's only connected to one place. So it is a list, but that list is always going to contain only one element in it. So current gets updated to graph of current of 0. Eventually, I'm going to return an empty string.join of the character list that I have formed. But as I go through, the idea is I need to add something to the character list. And if we look at, for example, if we look at, visualizing this for a real data set or a, collection of k-mers that we might look at, and we look at our figure again, one way I could start would be I could start with TA, and then I could move here and say, well, it's TAA, so I'm going to add an A, and then I add a T, and then I add a G, and so forth. So at every point in time, I add one symbol to the end of the string. That winds up being a little trickier than it needs to be, just because I would start the string as containing the first node, and then every time I enter a new node, I would add one symbol. But then the problem becomes when I wrap around and I've used all of the edges, I'm going to have extra symbols on the end, because then when I go, GTT, and then when I come back here, this is TTA, so I'd add another A, and so forth. But I now have wrapped around with the TA that I started with at the beginning. So it's a minor point, but it will just become an annoying algorithm if we think about it that way. And so there's actually a nice simplification of this, which is to start with the symbol T here, and then simply only add one symbol every time we go in. So when I move to the next place that I see, every edge, I'm actually going to only add the first symbol. So if you look at the first symbols that we see, we're going to have a T, an A, Then we have another A, and as you a T, a G, a C, an A, and so on. So as you go through this, every time you use an edge, I'm actually only going to use the first symbol of the edge. It's a clever way of viewing this problem. And that way, when I get to the end, when I enter, say, use this edge, I'm using only the G. Remember, I've used the T before, and then when I wrap back around, I'm going to use a T here, but that middle T is going to be the first T here. And now when I come back in, I have a T and then a T, and that's perfect because I'm not adding another symbol, and I've actually formed a cycle. So the clever idea is that I just add the first symbol of current to the character list. And so I will say chars.append just current of 0. And this is an optimization. Probably most folks, if they sat down and tried to solve this problem, they would do it the other way. They would start with whatever the initial node is and then keep appending symbols as they go to the end. But this is a nice tight simplification of it, where I just take the initial symbol of the current node at each point in time. So I'm still adding a symbol every time I enter a node, but I'm doing it in a clever way so that when I run this on the sample, I get our result. And we say, oh no, that doesn't match what we see on the platform because the platform said Gattaca. It still is Gattaca. It's just that we started at a node that didn't start with the G. So we started at the Attica, and then we have a G on the end, and that's the same string as a cyclic string. So the autograder will be fine when I run this with actually assessing that as correct. So at this point in time, we make sure that we've refreshed the page. All right, we're going to download a data set, and then I'm going to put that in the File Explorer. And I'm going to call it data set. And I also want to make sure that I comment out all of the parsing part of this code. And run it again. And copy in the output. And it suggests that we go on to our next question, which we're going to do. Substantially less drama on that problem than we had on the first one, because you can actually see my screen, which is nice. It's a nice change. And moving on. Henry, I'm glad you got the Gattaca reference. Yes, I saw Heyerholzer's algorithm exactly. Yeah, that's the not necessarily the most efficient approach for finding an Eulerian cycle, but it's definitely the one that's very teachable. And so that's the one I approached the problem with. But more on that in Great Ideas in Comp Bio. That's not the direction we're going today. All right. We'll get ready for the next problem then. Let me know if you have any questions. Next problem is going to be GASM. Okay. All right. The fatefully named GASM is genome assembly using reads. And the idea of the problem here is that, well, we've seen a warm-up version of genome assembly using a Bruijn graph, where we assume that we have perfect coverage. So every single possible k-mer gets sampled as part of our data set. That's obviously a very strong assumption. And there are two practical considerations that we need to meet head on and work with a little bit. So the first one is, well, what would you do if when you're generating all of your reads, you're not getting every single possible starting position in the original string sampled? This is a problem that's gotten better over time because, well, generating sequence has gotten cheaper and the experimental protocols and everything have gotten better. So it used to be the case that you might have a lot of regions missing from the genome when you were producing the sequencing reads. Over time, as sampling them has become more inexpensive and easier to obtain, that's become less of a concern. However, at the end of the day, assuming that you're getting every possible k-mer sampled from the data set is a strong assumption. What we're going to apply is a simple idea. The simple idea that we'll note is here on the left, you see a collection of k-mers, and here k is equal to 10 that have been sampled from a genome, and we did not capture every possible starting position of length 10 from the genome. So for example, we didn't get this substring of length 10 that started TGC and so forth. We did get GCCG, TAT, GGA, but we're missing one in the middle. The simple but good idea here is to note, well, yes, we didn't get every substring of length 10, but we actually did get every substring of length 5. So you could obtain a perfect coverage with k equal to 5 simply by splitting each one of the reads in your data set into all of its constituent substrings of length 5. In this particular case, ATGCCGTATG splits into ATGCC, TGCCG, and so forth until we also get GTATG. So one original read gives me, in this case, 5 substrings, and by making a smaller value of k, we're able to boost artificially the coverage of the data set that we have. There's a problem here, of course, which is that, well, we don't want k to become too small. And the reason why is a large value of k is very useful. You might imagine the reason why is, say we've got a repeat in our data set, and the repeat is of some length, but if we're able to see either sides of that repeat in the original read, that's very useful because it gives us additional context. It says, I have a repeated string here, yes, and repeated strings make genome assembly hard. However, I can see on either side of it, and that's going to help me overlap it and place it into the correct position in the genome. When I start breaking the reads, I might lose some of that information. So in the extreme case, the worst thing you could have would be to have a value of k that's equal to like 2, because then you're just obtaining, well, there's only 16 possibilities for a substring of length 2, and you're not getting very much information from the data set at all. So if you break the reads too much, you start to lose all information. What you would ideally have is a sequencing technology that would read an entire chromosome, like tens or hundreds of millions of nucleotides all at once. And then all of genome assembly would be solved. You wouldn't need to overlap anything at all. We live somewhere in the middle, which is that we're sequencing fragments, but sometimes those fragments are not going to give us the coverage that we need, and so we want to break them into even smaller pieces, but we don't want to overdo it. So we're trying to find a Goldilocks value of k, where when we split the reads, those reads wind up splitting into reads that cover the genome well, but that aren't so small that they become useless. So this read breaking picture is an illustration of the context that, well, in practice, you might have reads of length 200 and you fragment them down into length 80 or something like that. The other important consideration The other important consideration is, well, you have double-strandedness, and that's not something that we have discussed quite yet. But we've talked about forming the de Bruijn graph of all of the k-mers plus all their reverse complements. But we've not talked about, how do we address, what's going to happen when all this goes into one network. And you'll notice in this particular case, here's the de Bruijn network from all of the substrings of length 3 of this string. Here's the de Bruijn network of its reverse complement. So you can see in this particular case, it ends with CATA, and you can see we go in the opposite direction, we would get TATG and so on. So this is the de Bruijn network of chopping up the reverse complement into all of its substrings of length 3 and forming that network. In practice, you can see that we have highlighted these using red and blue. In practice, what you might see is, well, the combination, the amalgamation of these two networks. So if you had perfect coverage along both strands, you would be able to see this network that is quite a bit more tangled. And then the question becomes, how do we identify an assembly from this, given that you have k-mers coming from either place? You have them coming from one strand or another, and in some cases, they're going to mix and tangle your network further. So the idea of this question is you might not get perfect coverage of just the k-mers from one strand, but when you sample and you look at these coming from either strands, meaning you take the reverse complements of all of your k-mers, we're going to reverse complement those k-mers, break them down, and then find the point at which this problem becomes solvable. So if we look at this previous figure, the idea is we wouldn't be sampling all of these k-mers. We're going to be sampling some of them, and we're going to be sampling some of the k-mers over here. And what the problem is getting at is, can we use read breaking to boost the coverage, so we're covering the genomes better, and can we use the fact that we've got k-mers coming from either of these strands to at least appreciate this idea that both read breaking and taking the reverse complements of all of your input strings are both really helpful to try and make sure that you're capturing something that you can ultimately reconstruct, even though, as I said, it's going to make the problem harder. So our input here is a collection of reads of some length, and then we're going to form the de Bruijn graph, but we're going to form the de Bruijn graph by taking reverse complements, and we're also going to want to break our reads, because the idea is when we form our network, well, if we just use the input reads, it's not going to be possible to walk and get everywhere, actually be able to go from a starting point and form a cycle. There's going to be a point at which we get stuck, and we're going to get stuck because we can't see the next k-mer. So what we're going to do is we want to break these down so that it is possible to jump from one position to the next. But we want the largest value of k where that happens. So what the problem is trying to communicate is, it's like a toy problem, but it's communicating a very deep idea, which is, what value of k do you want? You want the biggest value of k when you break the reads as possible to ensure that you have good coverage. And this problem is going to say, we're going to give you a very nice data set where when you form the de Bruijn graph, it has exactly 2 directed cycles. So in that sense, we're going to have a cycle corresponding to the genome, and we're going to have a cycle corresponding to its reverse complement. And so what we're trying to do is find the point at which that becomes true. So we're given a very kind of strong assumption, which is that the data ultimately are nice. They're not quite as messy as what you might see in real practical data, but we're kind of preparing you for these ideas of needing to break the reads up into fragments and take reverse complements. So we want to form the cyclic superstring, and the idea is, well, it's Gattaca again, but one of the strings that we're going to encounter in our data set is going to be Gattaca, and the other one is going to be its reverse complement. So we'll have 2 walks through our network. One is Gattaca, the other one is going to be its reverse complement, which in this case is TGTA ATC. But we're ready to solve this. Our idea is going to be, we're going to range over all of the different k-mers different values of k that we might use, we're going to fragment all of our reads up into those k-mers that can be formed from them. So we're going to break the reads. We're going to make the de Bruijn graph from it, and then we're going to try and walk through that de Bruijn graph and find a superstring or form a cycle. And if we form a cycle that uses half of the edges in our original data set, that's good. That means that we have found a cycle that returns back to our starting point. We can check that too. It needs to return to our starting point, meaning it forms a cycle, and it needs to kind of have used enough edges in the network. If you don't satisfy those, we're just going to say that value of k doesn't work, so I'm going to try a smaller value of k, and then we're going to iterate the algorithm. If we don't find anything, we're just going to return an empty string. So our highest level function here is going to be called shortest circular assembly. It's going to take a collection of reads as input. It's going to return a string as output, and that string is going to be circular. And we're only going to return one because the idea is the data set's going to guarantee that there's another circular string here, which is its reverse complement. But we're not going to worry with that because the problem is telling us that the data are in this nice fashion. So our idea is to range over all possible values of k. Critically, this needs to be descending because the larger the value of k, the first value as we go down, the first one we find is going to be the one that we want to use. And as we range over all these values of k, we're going to basically try to form a superstring using the de Bruijn graph, and then we're going to return our first success. So I will assume that all of the reads have the same length. We could write an assertion function for this, but this is an invariant coming from the data set that we're going to be given. So I'm going to set a read length equal to the length of the initial read. Okay. And then once I've done that, I'm going to say, well, let's range over all the values of k. So for k in the range of the read length, and we're going to start it at -1. And then we're going to go down to 1, and we'll have a step size of -1. So that's going to allow us to decrease by 1 each time. The last value of k we're going to consider is 2. And I want to form all the k-mers. So I'm going to say the k-mers that I'm interested in is all of the k-mers of the reads of length k. And then my candidate string, candidate assembly, is going to be equal to, well, let's call another subroutine that's going to try assembling these genomes, or these reads, and not the reads, but the k-mers. Okay, so once we form the k-mers, this is the read splitting step, and this is the does it work step. And if so, then we're going to be done. So how I'm going to flag that is I'm going to say if candidate assembly, when we try that, and it's not equal to an empty string, so I'm going to use the empty string to indicate that nothing was able to be obtained, well, then I'm going to return that candidate assembly and we're finished. If we make it through all the values of k, we failed if we make it here. So we didn't obtain an assembly, and I just want to return an empty string in that case. So we'll flag that over all the values of k we didn't find anything, and that would be useful. So for a given value of k, the candidate assembly could return an empty string. This is returning an empty string if we tried it for all values of k, and we failed in that case. The easier function here to write is just all k-mers. So we need a function that's going to perform the read breaking, and that function is just going to take as input a collection of reads as a list of strings. It's going to take an integer k, and it's going to produce a list of strings at the end of that. So our idea here is to collect all of the distinct k-mers that are occurring in this particular data set. And the reason why that's important is we don't want to overdo it. So we're kind of assuming that we're not getting a lot of repeats in the data set. And if you look back at our figure, and you look at our read breaking, the reason why we're kind of assuming this issue away is, well, a string like GCCGT, it's present in this original read as well as in this original read. So we don't want to add it to the data set twice, right? We want to be able to just have one path that goes through the network. Similarly, something like, I don't know, GTATG, that occurs in 3 different places. And so we don't want to add it to our data set 3 separate times. So what I will do instead is, as we make our list, we're looking for the distinct k-mers. And so we'll use a set to do this. So I'm going to have a set that I'm going to call scene as a set of strings. It starts off as empty. And then at the end of this, I'm going to return the list that is obtained when I sort it. So that will just, produces a list in a nice form for the user. It makes it easier to test and debug, for example. And then in the meantime, we're going to range over all our reads, split them, and add to the set. So I will say for each read, let's call it R in our read set. Then we range over all the k-mers in R. And here's where we get to apply what I like to call the magic formula of bioinformatics, which is that if you have a string of length n, in this case it's of length of R, and you're interested in finding every substring of length k in it, well, there are n minus k plus 1 possibilities. And so if we're ranging over all of them, the starting positions range from 0 up to length of R minus k. And we're going to have a plus 1 because it's not going to be included. Okay, so really it's range from 0 up to length of R minus k plus 1. So now we see length of R minus k plus 1 total possibilities, but we don't need the 0. The current k-mer that we're interested in, of course, is R of I to I plus k. That's the substring of length k starting at position I. That's good. And what we'll do is we'll add this k-mer to our scene set. So what's nice about add, it doesn't add something that is already there. So we're not going to be having duplicates get added as a result. And that's just because we have this built-in set data structure in Python, which is super helpful. But once we do this, we also want to do the same thing for the reverse complement of R. So I want to say RC is what happens when we call the reverse complement of R. I've already imported that here behind the scenes from RUVC, this problem that we get to use over and over again. And then I want to range over all of its substrings of length I. So I'll do another ranging over all of its substrings of length k. The current k-mer now is the reverse complement starting at I of length k, and then I'll add this k-mer to the set if it doesn't already occur. And then, of course, at the end of the day, I return the sorted list of all of the elements of that set, and we're finished. So we've written 2 functions here, our highest level function and the subroutine all k-mers. We're missing a function that says, okay, for your current value of k that you divided the reads up into, does this actually give you a candidate assembly of the form that the problem is telling you it's going to be in? So the problem is telling us there is going to be a certain value of k where at that point in time, the Bruijn graph is going to have this really nice property that there's going to be one cycle that's going to be nice and perfect and correspond to the original genome, and then there's going to be one that corresponds to its reverse complement. So let's write that. This function is going to take my k-mers as a list of strings, and then it's going to produce a superstring, a circular superstring from walking through it. And so the first thing we might do then is, we need the Bruijn graph. And so I'm going to say graph is equal to the Bruijn graph of the k-mers. Notice when we try the assembly, we've already formed the k-mers of the reverse complements of all of our reads. So I don't need the Bruijn graph with the reverse complement too. So I just take our Bruijn graph function that we wrote in DBRU, and it's going to take my collection of k-mers and produce their graph. Then we want to make a check. So if any node has more than one outgoing edge, the graph isn't in our nice, pretty form that the problem guarantees. So that would be a problem. And I could produce an error message or flag this in some way. But what I will do is I'll just say, let's make that check, and we're going to return an empty string if we see it. So how is it that we check this? We need to range over all of the elements of our dictionary that stores this adjacency list. So remember, the graph is a dictionary mapping strings to list of strings. Okay, so each string is the starting node, and then it could be connected to one other node, or it could be connected to a million. And what we want to ensure is that it's only connected to one, meaning, well, what we want to range over is graph.values. So those are the values of the dictionary. What are the values of the dictionary? Each value is a list of strings. So the value that I'm ranging over, I might call this like suffixes, okay? So, but these are the things that each prefix node is connected to. And if it's ever the case that that list has length greater than 1, this would be bad. So this violates the condition of the data set, and I want to return an empty string, or produce an error. Hopefully we never hit this. Okay. But if we make it here, we know that the data set kind of has the form that we care about, which is every node is connected to one other node. So the only other thing that can go wrong is we need to check that we can form a cycle. That's the critical part here. So I'll do that greedily because, well, if we ever had more than one option, we would have returned an empty string already. So every node is connected to only one other partner, and I'll say this can be done greedily. Okay, so we're saving beautiful algorithms for exploring our Bruijn graph for other contexts and other days. But for now, we'll say our starting node needs to be somewhere, and we'll apply the same idea that we saw in a previous problem where we're going to take the first k-mer, and then we're going to take its suffix or its prefix. So the prefix needs to have length equal to whatever that first k-mer is, minus 1. So this is the prefix here, going up to and not including the final symbol of that k-mer. I'll do the same thing as before. I'll set current equal to start. So that's going to keep track of where I am in the network as I continue walking by using this, tracks the current node. And then the other thing I want to do is identify, what are the nodes that I have already visited. So I'm kind of checking things off of a list, and I'm going to use a set to represent that. So I'm going to call that set visited. It's a set of strings, of course, because these are the nodes. It's going to start off as an empty string because I haven't seen anything. And then the other thing I will do is, we need a character list to store the symbols that we're concatenating to form a final genome. So we're going to have chars is a list of strings, and it's going to start off as empty. At the very end, what we want to return is the join of that. So we could also just keep concatenating to a string, but this is the more efficient way of doing it, as we've seen repeatedly. Now we're ready to walk. So we are going to walk through the network until we hit something we've seen. So I will continue walking until, well, I form some kind of cycle. And that doesn't mean that I've hit my initial starting point, but it just means that I've come back to somewhere that I've already been. That means that at that point in time, I've formed a cycle, and that's either where we started, which is good, or it's something in the middle, which would be bad because, well, the data set tried to promise us that there's only one way of walking through the network and using each kind of one in, one out edge, and then eventually coming back to where you started. And so we'll use visited to keep track of that. So how is it that we know we've seen something? Well, while it's the case that the current node that we're at is not in this set of the visited nodes that we've seen, if I can spell it correctly. So while it's the case that the current node is not a part of that, then we're going to first apply the same trick that we saw in our perfect coverage problem, which is to add the first symbol of the current node to our growing list. So we're going to say this is allowing us to form the genome. We'll say chars.append of current of 0. But we also want to, check off that we have visited the current node. Okay, it wasn't in the visited set, so we need to add it to the visited set. So I'll say visited.add current. And now we need to check. So we want to say, step forward. And to step forward, I'll say update the current node to be equal to the graph of the current node. But remember, graph of current is a list of strings. We know that that list of strings can contain only one thing because we made that check. And so if it's a list that contains only one thing, we know that we need to grab what is present at its index 0. So we have graph of current of 0 is stepping forward. There is one other thing we probably should check, which is that as we walk through the network, it might be the case that we hit a node that doesn't have any edges emanating from it or incident to it. So we have tried to access graph of current, but it might be the case that graph of current doesn't exist. So I want to make sure that when I access this graph of current that it really is something, that I'm not at a node that just doesn't have any edges leaving it, because that would be another condition where the data set would if you think about read breaking, we've hit a point where we can't actually jump across to the next place. This value of k wasn't small enough for us. And so when that's the case, we need to say if current is not a key. So if it's not in graph, this is no edges coming from the current, and that would be bad. I want to return an empty string. It means that we hit a stopping point. Okay, so I would say k isn't small enough yet. So we need to carry on, and so I will carry on to the next value of k just by returning an empty string. After we continue doing that, so at this point in time, the while loop is over. So if the while loop is over, and we said while current not in visited, what is it that we know at this point in time? We know at this point in time that current very much is in visited, and so we have returned to a node that we've already seen. Okay, that's a good thing. If we're not back at our starting node, then we need to move to the next value of k. So I will check to say, is it the case that current is not where I started? And if it's not where I started, I want to return an empty string. But if I pass that check, now at this point I know that I'm at the starting node. And that's good. I had a cycle, and that's what I want. There's only one other thing that would be bad, which is imagine, well, you've walked through the network, but the network was supposed to be huge. It was supposed to have 100,000 nodes in it, but you just did a quick cycle where you kind of left your house, you turned around, you walked back in. So that the cycle only has like 4 edges in it. That would be bad. So I don't want to conclude that I've got a final answer here. We need to ensure that the cycle has the correct length. So I have a cycle every time I encounter a node. It's the first time I encountered that node. And then at the end of this process, I know I'm back at the start, but I need to make sure that the number of edges along the way is right. And so what I will do, and there's a lot of ways to check this, but I'm just going to say if the number of visited nodes is equal to half of, our check is the number of visited nodes is equal to half of the k-mers. So every time you use an edge, you visit a new node. You continue doing this. So I can keep track of the number of edges by just saying it's the number of visited nodes. And that's an easy check for me to make, because I could say, if it's the case that, the length of visited is equal to, well, let's do the opposite check. So if it's not equal to the length of the k-mers divided by 2, I know I have a problem. I make this a little bit easier by saying multiply the number of visited nodes by 2. If that's not equal to the length of the k-mers, well, this is not a valid solution. And so we're going to move on to the next value of k, and we have another place where I can just return an empty string. If we make it past all of this, at this point, we know that we have a valid solution for a lot of reasons, right? It uses, we have checked that we've formed a cycle. We're back at our starting node. Return to our starting node. We also know along the way that we entered each node exactly once. That was good. And it has the appropriate length. So we've satisfied all these conditions, and that's essentially what the data set is telling us, that we're not assuming a very hard type of sample data set, but we're assuming a number of conditions, and under those simplifications, if we follow these steps, then we're going to have a valid solution. And we can try this by running Python 3 GASM.py. And you might be concerned because, well, we know that the data set that we want to have in main is Gattaca. And you might look at this and say, well, that's not Gattaca. I can look at all the different rotations of it. But remember that the data set is going to produce 2 cycles. It will produce a cycle corresponding to Gattaca or its reverse complement. And if we look at the reverse complement of that, it's TG, T, AA, T, C. And we see TC, TGT. I was about to be worried. TGT, AA, TC. So the autograder here is doing a lot of work. It's saying it can be any rotation of this string, or it could be any rotation of its reverse complement, and that's okay. So I think we should be comfortable in commenting out or uncommenting out the multi-line comment that we have, downloading a data set, putting it in our file explorer. We're going to rename it dataset.txt, and we're going to run our code again and grab it from the output. It's very fast, which is nice. And then we're going to take this back and put it into our submission. All right. Oh, we got a lot of badges. Look at this. And we unlock 2 problems, which are going to be the next 2 problems that we look at. Pretty good. So we got a genome assembly badge. That's nice. Hey, puzzles. We got a badge for being good at a lot of topics. At this point in time, I would hope we would get that because we've worked on a lot of different areas. Yeah, and we've also looked at a lot of graph algorithms. So good. And here's the bridges of Königsberg, which is the famous problem where Eulerian paths were first sort of identified as walking through a city, trying to cross every bridge exactly once, and returning to your starting point. So if you check out our bioinformatics algorithms book, we talk about this as the motivating question from 18th century Prussia, and that there isn't an algorithm for starting somewhere in the city, using every bridge exactly once and returning back to where you are, and connecting that to genome assembly where the problem does have a solution. Okay. 2 more questions today. GREP is going to be our next one. get ready here. Any questions, let me know. Otherwise, I'm going to move on to our next question, which is, I think, probably the most challenging one of the day. All right. GREP is genome assembly with perfect coverage and repeats. The idea of this question is, you may remember, or you may not, that the greatest complication in genome assembly problems is repeats. So we have our repeats make jigsaw puzzles hard to solve. So we look at our Eternity 2 puzzle, which had no known produced human solution in 3.5 years, despite a $2 million cash prize. And that was because of the repeated information present along each half of one of our puzzle pieces. But we also could look at one of our real networks for a collection of sample k-mers. And we're going to see the same sort of repeated structure causing issues too. So if we look at this de Bruijn graph that we have for an example collection of substrings of length 3 that come from our bioinformatics algorithms book, it's not the case that every time you enter a node, you have only one option for leaving that node via an edge. So when we enter TG, for example, you could go north, east, or south. It's not immediately clear which direction to go. And the reason why this complication is introduced is because of repeats. So this TG occurs in a few different places in the genome. Sometimes it occurs before a C. In this case, it occurs before a T. And in this case, it occurs before a G. And so it's not clear when you arrive at this node for the first time which direction to go. So it's the same exact phenomenon as when you look at a puzzle piece. It's not clear which of the possible options for that puzzle piece to choose to place at this particular point of the puzzle, which is what makes this complicated. What this problem is getting at is to say, well, at least for small data, can we form all of the circular strings that arise from valid cycles in the de Bruijn graph? Because it's going to turn out, and what the problem is kind of getting at, that you may be able to construct example collections of sequencing reads that could have produced more than one string. So the input here is going to say, I've got a list of DNA strings, and let's just look at, we're not going to consider read breaking. We're kind of taking a look at different aspects of the problem that can be complicated. If we try to put all of them together in one place where we're going to have read breaking, we're going to have reverse complements, and now we're going to have multiple solutions, this is going to become very challenging for us to have autograders around checking this. And we're not trying to overwhelm the user here. We're just trying to expose them to these considerations in this independent problem solving website to give you an opportunity to delve deeper if you like. But we're taking everything from some circular chromosome, and then we want all candidate solutions. So we're going to make the de Bruijn graph of those collection of reads, and then we're going to find all of the valid cycles in it that could correspond to a solution. So the idea here is to see that you could have one collection of k-mers that actually could have come from multiple different strings. So there are multiple different strings behind the scenes that when you chop them up into k-mers, you wind up with the same data set. That might not be something that you would anticipate immediately, but it's something that is true. And this problem is saying, well, if this were your data set here, this is a collection of substrings of length 3, you actually can obtain, you'll notice that they have similarities. Why? Because they have the same substrings of length 3. So they're ultimately going to have to be similar strings, but notice that they all start with CAG, for example, CAG-TT, but then they have slight disagreements. They all end with TT. They have slight disagreements kind of in this interval, and that is caused because you have repeats that you can see that are present in the data set. Okay, so TT, for example, is a substring of length 2 that you see quite a bit, right? And that's kind of going to cause some issues because when you enter GTT here, well, maybe the next symbol that you see is a C, maybe it's a T, and maybe it's a G, right? And so that's what's causing us to, at the end of the day, see multiple solutions for this. So the problem is asking us to do something very precise. What the problem is asking us to do is to make the de Bruijn graph and then walk through the de Bruijn graph and find every possible valid Eulerian cycle, meaning touching every edge exactly once in the graph. That seems like it could be a huge problem. And in fact, if we constructed a huge data set with many, many candidate solutions in this and tons of repeats, it's going to be hard to have a very efficient solution. Fortunately, that's not really what we're going to check. We're not going to produce a solution for this problem that is perfectly optimized because it's going to be challenging enough as is. And so what we are going to do is produce a reasonably fast solution that's nevertheless able to obtain every Eulerian cycle in a network. So we're doing something that seems like it has this biological connection because it does, but what the problem is really asking us to do is to take a network and produce every single possible Eulerian cycle in that network. So let's do that. Our function is going to be called all circular assemblies. All it's taking is a list of substrings of length k. It's not going to return one string, it's going to return a list of strings, which means maybe that list is empty, maybe it has one thing, maybe it has a million things in it, we're not really sure. Our solution, which I said is not going to be perfectly optimized, but still is going to be good. Our solution is going to be called depth first search here, which makes sense. So at the current node, we'll pick an edge leaving it, recurse to find all paths along that edge, put the edge back, and try other outgoing edges. So over all of the possible, I could have 10 neighbors of this current node. I'm going to explore in each of the 10 directions. And when I go in this direction, I will explore every possible path that comes from that direction. If I find any Eulerian cycles along that path, great. I'm going to remember them because at the end of exploring along that path, I'm going to move to the next edge and I'm going to explore there. So the idea is I might have 10 neighbors. Some of those neighbors might eventually produce an Eulerian cycle. Some of them might not. And I'm going to combine all of them at the end. So the first thing I want to do is make the network, and we will say graph is equal to the de Bruijn graph of our k-mers. Each Eulerian cycle will, by our convention, start at the first node. So I'm going to have a first k-mer that I'm going to declare as k-mers of 0. And I need to say, well, the starting node is not quite that. The starting node is whatever that first k-mer is, but everything up to, but not the final symbol. So it's the prefix of maximum length, or length k minus 1 of that string. And the second node that I know I'm going to look at is going to be, well, the suffix of that, of length k minus 1. So I'm going to start at position 1 of the first k-mer and go to the end. So the idea is recursion. So what we need to do is we need to keep track of the number of edges so that I know if I've used them all, plus keep track of the completed assemblies. that we found. So there are two extra kind of, I guess, bookkeeping, for lack of a better word, variables that I'm going to declare. We're going to declare total edges. And what is that? Total edges is the length of our k-mers. Each k-mer in the data set corresponds to an edge when we make the Bruijn graph. So the length of the k-mers is the number of edges. And the assemblies that I want to eventually produce are going to be a list of strings, and that list of strings is an empty list. So once I've got this bookkeeping, then the next thing I'll say is, I've now used this edge. Get rid of the edge that I've used. So I'll say, I'm going to look at the graph, I'm going to look at the start node, and I have a remove method that Python gives us, which is good. So if you have a list, you can just call remove and get rid of the string here, which is second node. So we're getting rid of that edge, in other words. Remember that in this graph, it's a dictionary that maps strings, which are nodes, to lists of strings. And so those are the nodes that they're connected to. And I'm removing this particular edge connecting start node to second node. So now we recurse with a function that has, I think, roughly 1 million arguments. Okay, and hang on to this because I'm going to explain all these arguments. And you wouldn't be able to just magically know all of the things that you're going to put into this function, but I'm going to put into this function like everything I can imagine. So this function is going to be called extend. This is going to essentially take my network, take the starting node that I care about, and then form all assemblies of that network starting at that node. So really, it's going to take the graph, it's got to take second node, and it's got to take the completed assemblies. The completed assemblies are going to grow. So you might imagine we could have written this in this way. Okay, but we're not going to need that because this is a list of strings, and I'm just going to update it in place. So every time I find a completed assembly, it's just going to keep updating this list, which is a parameter because, well, lists are essentially passed by reference in Python, so I can update this list in place. Technically, we need to remember a few other things. So graph, second node, and completed assemblies are good, but I'm also going to have some conditions as I go that make sure, so this function needs access to other things. So for example, to make the checks and know that it's found a solution. So one of the things it needs to know is the number of nodes that it's encountered thus far. So at this point in time, it's encountered one node thus far. The other thing that it needs, and I'm getting the order a little bit out of whack here, but the other thing that it needs is how big is what is the path that it's encountered. So at this point in time, we're only actually going to be able to conclude one symbol of that path, which is the first symbol of the start node. So this is our growing string. This is the number of nodes that I've encountered up to this point in time. And then we're going to have the number of edges in the network. Why is that necessary? Because the network here is maybe going to remove edges as we go and so forth, but at the end of the day, we're going to be done when this is equal to this. And that's probably not the case yet because our network is going to have more than one edge. But as we keep calling the subroutine, we know we're going to be finished if we've used every edge that we encounter. And I think I have start node. It's going to also go in there too. Why? Because I'm not done if I've used this number of edges. I'm done if I use this number of edges and the current path that I'm on ends at the start node. So there's just an enormous amount for this function to have to manage and juggle and keep track of. The recursion is going to be beautiful because this looks very like how this function is going to be hundreds of lines long. Actually, no, it just needs a lot of parameters to check. When it is done, at this point in time, if the recursion works, the recursion works, but the recursion is finished and we're done. So after we have called that and it's going to continue recursing, I'm just going to return completed assemblies. If I want to be nice to the user and have something that's easy to debug, I'm going to also sort them, but that's not required. Then I want to write the extend function. And as I said, I tried to explain these inputs to extend. It has a lot of inputs. It takes the network. It takes the current string that we've started forming. It takes current is always the node. Current is the node we're at the current moment in time. It takes the assembly that we've built so far. Okay, so let's look at those three things. This is the network. This is the current node we're at. This is the assembly that we have thus far. It only contains one symbol, but it's the first symbol of the first node at this point in time. Then we're going to have these two next things are going to say the number of edges I've used and the total number of edges, because I want to check that those two things eventually become the same. So I have the edges used and the total number of edges, both as integers. Then we're going to have the start node and the completed assemblies list that I'm growing. So the start node is how I'm going to check whether or not a solution actually comes back to its starting node. And then I've got the completed assemblies that I continue updating. So that's good. And everything that's going to be passed by reference. So I'm going to update it in place. And those are all of the parameters that we need. Note that we're going to return none again, because why? The action that it's taking every time through, if it's doing something, is just updating the completed assemblies. So we've got this function to write, and then the question becomes, what are the base case and the inductive step for this particular problem? That's really important. Why is it that recursion is helpful in this case? And it's because, well, I'm in a network. If I'm thinking about applying this depth first search and I want to find all possibilities, I can break that down as to saying, if I have 10 neighbors, I can try this on each of the 10 neighbors and let them recurse in their direction and return back whatever they find. But I have subcontracted out the work, the critical central concept of recursion. I have found a way to subcontract out this work by exploring in each of the 10 directions that emanate from me. So the point becomes, the inductive step actually might not be as hard as we think, but then how is it that we know that we're done? The base case is going to be, we've used every edge and we're finished. So we need to check two things. So if we've used every edge and we're back where we started, we're finished and we want to update completed assemblies. So this is an easy check. This is why having all these parameters is useful, because I'm going to say if the number of edges that I've used is equal to the total number of edges in the network, and I'm back where I started, meaning that the current node that I'm at is equal to the start node that I'm passing in also as a parameter here, well, great. That means I've formed an Eulerian cycle, and I'm going to append to the completed assemblies this partial. Partial is the assembly that I've built so far. It's just a string. And I'm finished on this in this direction. So I don't want to continue doing anything. I'm not returning a value, but I am going to return. Okay, so this essentially just says don't continue in the function. We're also going to have another base case, which is really just kind of a check. And this is a dead end. So if the current node, I keep typing out what I'm saying, if the current node is not in the graph, meaning no edges leading from me, well, I'm going to return. Okay, so I don't need to update completed assemblies. I've hit a dead end, and this function is finished. It's not going to need to call any subroutines recursively because there's nothing to call. The inductive step, the engine of recursion, is range over all the neighbors of the current node and apply the recursive call. So I will say neighbors is equal to whatever the list of strings is corresponding to looking at the graph and accessing the current node. And then we're going to range over them. So I'm going to say for the next node that we're looking at in the neighbors, And I might have like, there's one very minor thing here, which is I might be connected to a lot of different neighbors. If for some reason 2 of those have the same label, there's no reason to range over both of them. So I'm just going to say in set of neighbors. That probably doesn't make a huge amount of difference in this particular problem, but I want to do it for the sake of correctness. The idea here is I'm now looking at an edge. So I'm at the current node. Next is telling me what my next node needs to be. And what I want to do is to say I've now used that edge, so to speak. So I want to remove it from the neighbors, meaning, remove this edge. Okay, it looks like I'm removing a node, but I'm removing the connection from the current node to NXT. So I'm removing this edge, and the reason why is I've used it. So I want to remove it from the data set, but I'm going to recurse, and then I'm going to add the edge back. And the reason why is I still want this edge to be accessible on another path. It might be that one of the other neighbors needs to come back to me and then use that edge. So this is a very minor but very important point that I add it back after I apply the recursion. When I apply the recursion, this edge needs to have disappeared from the data set, from the graph, and so I will call extend. And now it's just a matter of saying, oh, this seems like a nightmare, right? But not really, because what are the parameters? The parameters are the graph. Okay, well, the graph has been updated to get rid of this neighbor. So the graph is 1 edge smaller. And after I do that, I've got the current node. Okay, well, I've updated the current node. So the current node that I care about is this neighbor that I called next. Then we say, okay, as I move to the next node, I have my partially formed genome assembly. Fine. So that's wherever I have currently, but I'm going to add plus current of 0 because that's where I am with the current node. So I'm adding 1 symbol to it. And yes, I'm doing string concatenations here, mea culpa, but that just makes this a little bit easier. I could generalize this, feel free to a list of strings and then concatenate them at the end. The next parameter is the number of edges that I've used. I just used an edge, so that's going to go up by 1. The total number of edges is not going to change, so that's just going to keep getting passed in without getting modified. So I'm going to say edges used plus 1 total edges. And then there's 2 other inputs. There's the start node. That has also not changed, and the completed assemblies is what I've updated along the way. So start node is the same, and completed assemblies is the same. And it's a little bit bizarre because, well, at the end of the day, that's the function. It's kind of like how it often is the case with recursion is I've actually done everything. I have my base case. If I reach a dead end, I have that covered. If I have a solution, I have that covered. And in the meantime, this process is going to either use all the edges or hit a dead end. And the recursion is subcontracting out all of the work that we care about. So the function is technically finished. Let's make sure that there's no issues with it. So I'm going to run Python 3 grep.py. And of course, I'm going to have an issue here because I think I named something the wrong thing, line 122. None type is not iterable. That's not good. That's in main. Okay. So I think this means I'm not actually getting anything. Yeah. So we're printing none. So we don't have any solution as of yet, which means that I need to take a look here. Made the Bruijn graph. This all looks good. I don't think this is going to make any difference whatsoever. At this point in time, it did. Oh, Python. So I call dot sort instead of sorted. I guess it just didn't like that. Okay, good. Good news. Here's the solutions that we had before. And we're in good shape. All right. So we've downloaded a data set. Let's get rid of this comment. And I'm going to name it dataset.txt and we'll run it again. All right, got an answer. Might be wrong, of course, but we're going to put it in the platform and see. And we got it right. All right. We're now person 306 to solve that. And it's a tricky one, not because it's long. I mean, we've written much longer code than this, but that DFS is a challenge to get right. And to optimize it, it actually really makes for a complicated solution. Note, this by far is sort of the hardest problem I think that we've seen. It's pretty deep in the problem tree, but only 300 people have ever solved it, so we're happy to be 306. Let me know if you have questions. I'm going to move on to the next one here, which has not been solved by many people because you actually have to have solved this genome assembly question first. But more people have solved it because it's not a hard question. So we will end today with a short question, which is nice because, well, we started with such technical difficulties that I'm still not sure where they came from, but we'll work on that for next time. All right, dataset.txt and output.txt are gone. This is going to be ASMQ. Should have named it ASMR, but it's not. I don't think it's a good enough problem for that. Okay. All right. ASMQ is assembling assembly quality with N50 and N75. And I should explain what those are, as well as why we care. So the idea is, well, we've talked about how to start thinking about the problem of assembling genomes. One of the questions that we might ask is, I could have an assembly algorithm, and then I want to say, how do I know if it's any good? One of the directions I could go is correctness. And that's not actually too hard of a problem to address because, well, you could take a known genome and chop it up into simulated reads and then plug it into your software and then see how accurately the software reproduces the genome. So that direction is not really the direction that we want to go. The direction that we want to go is quite a bit more practical. And the practical direction that we have is that every genome assembly algorithm doesn't usually produce one contiguous sequence for every chromosome that it's trying to produce. These days, for things like viral or bacterial studies, the database is large enough to consult against, and the assembly algorithms have gotten so good that some of the time, yes, you'll actually be able to obtain a final, essentially correct sequence corresponding to that chromosome. But still, mostly the reality is for eukaryotic studies like ourselves and kind of experiments that aren't trying to spend a fortune on sequence. At the end of the day, what you're going to obtain is some regions that you're confident about, and then there are regions between them that you're not as confident about, that maybe you didn't get enough sequence from. And so what you're obtaining at the end is a collection of contigs, contig being short for contiguous. So a contig is a contiguous stretch of the genome that you have reconstructed that you feel confident about. So it might not be that you're missing everything in the regions between these contigs. It might also be that, well, you had some sequence there, but you weren't quite as confident about it, so you weren't able to report it accurately. And we want to make sure that when we do report anything associated with the assembly, that we're confident about that assembly. So at the end of the day, we're getting contigs and not necessarily full chromosomes. And so the question becomes, well, how do we report how good this assembly is? As I said, one direction is to talk about correctness. The other direction is to look at the contigs themselves. And I don't think it's a complicated idea to say, what is it that we want in the contigs? Well, we want the contigs to be long and cover the genome. One thing that would be bad is if our contigs have 100 times the length of the original genome. So that's not ideal. But assuming that that's not the case, we want long contigs, and we also want to have not very many of them. And it would be nice to have one statistic kind of combine both of these ideas, right? Like this is something that comes up, I think, generally in the field of data science or data analysis, that I have kind of two or multiple different competing concepts or statistics that I could use, or metrics. And then how is it that we can combine them into one thing? One way that we can combine these two notions of having few contigs and ensuring that they're sufficiently long to cover the underlying genome is called the N statistic. And here's a figure that shows the N statistic, like in the most typical value of the N statistic, which is 50. The N statistic takes a percentage that you care about as a parameter. And what it returns is a number. So if your assembly size, your underlying genome that you care about, is, say, 1,000 million base pairs. So you have 1 billion base pairs in your underlying data. What you do is you order your contigs in descending length. So you might have the longest contig is 93 million base pairs, then 87 million, and so forth. And then you track up to the point where you cover half of the length of the underlying assembly. So in this particular case, you add up all these lengths, and eventually you hit the point at which you hit the midway point of the data set. So you've covered half of the genome, and then you simply report the number 44 million, because that is the length of the contig after you've ordered them in descending size, where you finally reach the midpoint. This problem says we'll give you can imagine what N75 is. You reach the point at which you're at 75% coverage, and I don't know, maybe this is 11 million. Okay, and that would be the number that you return. We have a much simpler sample here, where you have a sample data set. You have strings of length 5, 6, 7, and 9. You add all those up and you get a total length of 27. And then you say, well, how many of these, if you have them in descending order, how many of these are necessary to get you to half of 27? So you would need to get to 14. Well, if you take the one of length 9 and the one of length 7, that gets you 16, which is good. That certainly gets you over the halfway point. So you would report 7 as the length of your value of your N50 statistic, because that's the length of the contig that gets you over the finish line to halfway. 3/4 of 27 is a different number. So we're looking for essentially 21 or 22, and we can get there with 9 plus 7 plus 6, which is 22. So we can cover 75% of the underlying assembly with everything up to the contig of length 6. We don't need the contig of length 5. So our N75 statistic in this case is equal to 6. The point becomes, it's a clever statistic, and if nothing else, the problem gets you to understand how this statistic works. And we can understand how it works by implementing it. The solution here is not particularly complicated though. The idea is, well, we're going to compute the total assembly length. We're going to sort the contigs in descending order. We're going to figure out the threshold. And then we're going to go through the sorted lengths, adding each one to a total until we hit the threshold. This algorithm that we're applying is like an exact one-to-one replica of how you would do this by hand. You'd say, well, what's the total length of all of your contigs? That's the first thing that you want to do. Okay, that's not hard. I have a total, it's equal to 0. I have a contigs list of strings as input, and then the percent that I care about, that's going to be an integer instead of a decimal. So we're taking that as an integer. But we're going to range over all the contigs, and I'm going to say whatever that total is equal to itself plus the length of the current contig. So I've got that value now. Total is going to store it. Now I want to sort the contig lengths in descending order. So I'm going to form a list, I suppose, of integers called lengths. And then I want to say for C in the contigs, I want to append the length of that current contig to the lengths. Okay, the problem is they're not in descending order, so we need to place them into that descending order. And we can call a sort function to do that. So we could say lengths.sort on that integer list. So now they're increasing, so we need to reverse them. So then I'm going to say lengths.reverse. And we could do that in one step, but I like saying sort them first and then reverse them. We're not trying to have the most optimized imaginable function here. Then we care about the threshold that we care about. Now, if you tell me that 50 is your input threshold, okay, that's good. So I want to divide it by 100 first. So fraction is equal to my percent that I take by 100. Okay, and so in that particular case, I would get 0.5, or I'd get 0.75 if percent is equal to 75. And then I want to multiply that fraction by the total number of lengths. So the threshold then is equal to the fraction times the total. And, you know, that's easy to understand. Like, for example, before we had, you know, the total was equal to 1 billion base pairs. And the fraction would be equal to 50 divided by 100, which is 0.5. And so the threshold is equal to, in this particular case, 500 million. Probably should comment it. So the idea is once we've got that threshold, this is a number of nucleotides that we're trying to exceed. So we're going to go over all of the sorted lengths of the contigs. And because we're going in descending order, we're going to add each subsequent one that we see to a total until I eventually like hit or exceed the threshold. So I'm going to have something that to keep track of this. That's going to say covered is equal to 0. And covered is just keeping track of the number of nucleotides corresponding to this sum of contig lengths. Let's do sum of, I've already got lengths. Let's do covered, that's fine. I'm going to range over all those lengths. Remember that they're now sorted in decreasing order. So I will covered plus equal to length, okay? And then I'm going to make my check. I'm going to say, is it the case that this current value of covered is at least equal to the threshold? As soon as that happens, I want to return the length because I want, you know, this is the length of the current contig. So that's the length of the contig that gets us over the threshold. And the idea is the bigger that value, the better off we are. That means that we have very few contigs and they're pretty long. If that value starts to get really small, it means that I need a bunch of small contigs to cover half or 75% of the genome assembly. And that's not good. It means that the quality of the assembly is petering out. So even if I have multiple contigs, it's okay as long as I can cover a substantial part of the genome with long contigs. And that's what this is getting at. So everything here is good. The problem is, well, technically we might not have crossed the threshold. That would be bad. And there's going to be a default value that we have, which is just to return 0. But largely we should be good to go. So we can run Python 3 ASMQ.py. That's giving us the 7.6 values. I'm glad those aren't reversed on the input data set that we had here. And then we can just comment out or uncomment out our parsing part of our code. We can just get a data set from the platform. Call it data set, run it again, and we get 122 and 92 as our answer, which I hope is our answer. And we're going to submit. All right, Rosalind was a little slow today, but that's okay. We'll forgive the platform. We get a correct answer there, and we're good to go. So that is it for today. Thank you for joining me. If you stuck around for the whole stream and you got through our technical difficulties, well, you're my favorite learner of all time. But for now, that's all we want to cover with respect to genome assembly. I think we've essentially solved all of the pure genome assembly questions on the platform. As I said, there's lots more to learn about genome assembly. I would encourage you to go out and learn more about it if this is something that you find interesting. You can check out our bioinformatics algorithms textbook that has an entire chapter devoted to it. But just because we're finished with genome assembly for now on this set of streams doesn't mean that we're done with networks. And so our work is far from finished when it comes to our work with graphs. But that's all for today. Next time, we're going to look at another one of these applications of graphs, which is drawing evolutionary trees, or not just drawing them, but actually, producing the algorithms that will take in some biological data set and produce an evolutionary tree, meaning a network of nodes connecting to edges that represents that phylogeny of organisms. But that's going to be the subject of its own stream, in fact, probably more than one. So I'll look forward to having you join me for that. And that's it for today. So until I see you next time, thanks so much.